Hello and welcome to the lecture on Japanese encephalitis, the killer disease that silently wreaks havoc on a global scale. Its impact is particularly devastating in Asia, where majority of the deaths occur. Despite its massive toll, it largely remains unknown to many. Despite its name, it is not exclusive to Japan. Now, let us look into the historical background of the Japanese encephalitis virus. The history of Japanese encephalitis dates to the 19th century when it was first recognized in humans and horses in Japan in 1871. An epidemic occurred in Kyoto Prefecture, and by retrospective speculation, it was regarded to provide the first description of the Japanese encephalitis. Later, in 1924, an epidemic occurred and there were several thousand cases with a mortality rate in excess of 60% that created considerable attention and concern. From then on, approximately every 10 years, a severe epidemic took place in Japan. The causative agent was first isolated in 1933 from brain tissues obtained from 5 fatal cases originating from the 1924 outbreak by Michitoma Hayashi and inoculated into non-human primates Macaca cyclopus. Japanese encephalitis was initially called Japanese B encephalitis to distinguish it from von economoencephalitis, also known as encephalitis lethargica. The transmission cycle and involvement of pigs and birds as reservoir hosts were elucidated in 1959, though the acknowledgement of Japanese encephalitis as a mosquito-borne disease had already taken place in the early 1930s when the agent was isolated from a Culex mosquito. Now, let us look into the timeline of occurrence of Japanese encephalitis cases in India. In 1952, first evidence for Japanese encephalitis viral activity was recorded by erstwhile Virus Research Center, which is currently known as the National Institute of Immunology, during their sero surveys for arboviruses. In 1955, first human case of Japanese encephalitis was recorded in Tamil Nadu. In 1973, first outbreak of Japanese encephalitis was recorded in Bankura and Burdwan districts in West Bengal. In 1976, a repeat outbreak occurred in Burdwan. In 1978, there was widespread occurrence of suspected Japanese encephalitis cases. A national level monitoring program was initiated by the National Malaria Eradication Program in 1978. In 2003, a national program for prevention and control of Japanese encephalitis was launched. In 2005, a major outbreak of Japanese encephalitis occurred in Uttar Pradesh. In 2011, first-time cases were reported from Delhi. In 2013, first-time cases were reported from Tripura and Arunachal Pradesh. Global distribution of Japanese encephalitis virus. Japanese encephalitis is confined to Southeast Asia and Western Pacific regions. Approximately 68,000 cases of Japanese encephalitis are reported annually, with 13,000 to 24,000 deaths. Currently, more than 3 billion in 24 countries are at risk of Japanese encephalitis infection. Epidemiological and genetic studies have reported expansion in the geographical distribution of different genotypes and JEV emergence in the non-epidemic regions. Over the years, the virus has spread from Asia to Europe, Australia, and Africa. The Japanese encephalitis virus genotypes. Japanese encephalitis virus is reported to have originated from an ancestral virus in the Indonesia-Malaysia region, which diverged into genotypes 1 to 5 and subsequently spread throughout Asia and Australia. In 1995, genotype G2 demonstrated to spread outside of Asia with widespread activity in the Torres Strait region of Australasia for the first time. Approximately 5 years later, a new genotype, genotype 1, was isolated from sentinel pigs and mosquitoes found in the same area as the previous outbreak. Genotype 5 virus was isolated in Malaysia in 1940s and then went undetected until 2009 when it was isolated from a pool of mosquitoes in Tibet and then again in 2010 in the Republic of Korea. Genotype 5 is not a common genotype, with only 3 isolates having been detected. However, the question arises if the re-emergence after so many years is indicative of genotypic shift in the area. In addition to the geographic spread, changes in molecular epidemiology of Japanese encephalitis virus have occurred throughout Asia. Until 1990s, genotype 3 was the most dominant genotype in Asia. However, surveillance data revealed that genotype 1 gradually replaced genotype 3 as the most frequent genotype in many Asian countries. Sequence analysis revealed a few variations in the genome that may have played a role in the phenotypic change. Genotype 1 has higher infection rate and shorter intrinsic incubation period than genotype 3. Other host and environmental factors such as effects of immunity of a population to different genotypes, changes in farming and animal husbandry practices, changes in migratory pattern of birds may also have contributed to the emergence of genotype 1. The Japanese encephalitis virus transmission. Japanese encephalitis virus is a dual host virus which is transmitted horizontally between arthropods and vertebrates. It is mainly transmitted by Culex mosquitoes. Pigs act as amplifying hosts, bringing viruses closer to human dwellings, particularly in Asia where backyard farming is common. Pigs, both domestic and wild, are considered the most important Japanese encephalitis virus reservoir because there is an increased incidence of natural swine infection. Furthermore, they develop high titers of viremia when infected, which last for 2 to 4 days. Culex mosquitoes tend to feed on pigs in nature, and the number of susceptible pigs is large due to a high turnover rate in the pig industry. Though usually asymptomatic, Japanese encephalitis virus can cause reproductive disease in pigs with losses such as abortions in sows, stillbirths or mummified fetuses, and reduced fertility in boars, reaching up to 70%. Newborn piglets may have clinical neurological signs such as tremors and convulsions, with mortality in non-immune infected piglets reaching up to 100%. Non-pregnant females may have mild clinical signs, fever, or subclinical Japanese encephalitis. Naturally infected pigs have long-lasting immunity and therefore are no longer able to amplify Japanese encephalitis virus, and mortality rates in adult pigs are nearly zero. Bird-associated transmission cycle. There are over 90 bird species, both wild and domestic, that can become infected and transmit the Japanese encephalitis virus. Once infected, birds become immune to Japanese encephalitis virus and are no longer able to amplify the virus. However, migration or hormonal stress could lead to a reactivation of latent Japanese encephalitis virus in birds. Wading birds of the Ardidae family, such as egrets and herons, are highly susceptible to Japanese encephalitis virus infection, reaching high viremia titers that last as long as 4 days and represent an important source of infection for mosquitoes. Vertical transmission between Mosquitoes and their offspring has been suggested as a potential strategy for Japanese encephalitis virus to survive the cold season and restart the transmission cycle every year in the northern regions where an epidemic pattern occurs. Besides vertical transmission in mosquitoes, the virus survives the cold season by overwintering in hibernating mosquitoes, mosquito eggs, or reptiles. Besides pigs and birds, other domestic animals that may have subclinical disease but likely do not contribute to Japanese encephalitis virus transmission are horses and other equids like donkeys, cattle, sheep, goats, dogs, cats, chickens, ducks, sylvatic mammals, reptiles, and amphibians. Japanese encephalitis in humans. Humans are considered dead-end host as they have a low level of transient viremia, and though they do not transmit the virus, they can become infected. In endemic areas, children and travelers are usually those who become infected. The highest attack rates occur in children between 3 to 6 years and become increasingly lower after 14 years of age, the time at which the levels of neutralizing antibodies rise, either by natural exposure to Japanese encephalitis virus by subclinical infection or vaccination. The epidemiological triad of Japanese encephalitis transmission. The classic epidemiological triad is an important tool in epidemiology used to describe the causative nature of the disease. It consists of the agent, the host, the environment, which are portrayed as vertices of an equilateral triangle. Disease results when three sides connect. Conversely, disrupting a connection using control measures such as vaccination, habitat manipulation, reducing host exposure, etc., will reduce pathogen transmission. However, the existing epidemiological triad paradigm is insufficient to explain the complex biology and interaction of arthropod transmitted diseases or pathogen transmission between hosts that is dependent on the arthropod vector. Often, in outbreak situations, vector control is one of the first and foremost effective measures put in place to reduce pathogen transmission. The three components of the epidemiological triad are agent, host, and environment. Agents, the first component of the epidemiological triad, are usually pathogens. Hosts, upon exposure to the agents, can become infected, although infection does not always result in disease because the host and the environment conditions may not be conducive to disease. Infected hosts can either amplify or carry pathogen for extended periods of time, referred to as the reservoir hosts, or increase the infectious particles but not for a longer duration of time, such as the amplifying hosts, or do neither, the dead-end hosts. Specific risk factors affect infection and severity of the disease, such as host exposure, susceptibility, and response to the pathogen. These are influenced by environment, the third component of the triad, which can include social characteristics such as socio-economic status, lifestyle, employment behaviors, and psychological, physiological characteristics of the host. Additionally, the environment can consist of biotic or abiotic factors outside the host, including habitat, geographic location, living conditions, weather, and climatic conditions. The mechanism by which Japanese encephalitis virus transmission takes place are related to a change in land usage and agricultural practices. As economy improves, and the rice industry flourishes at the expense of deforestation, increased opportunities for mosquito breeding develop. As rice paddy fields are considered ideal breeding grounds for mosquito development, paddy fields also attract migrating birds, adding to the complex interplay of factors characterizing Japanese encephalitis transmission and spread. Higher humidity rates and temperatures lead to mosquito development, and consequent higher number of vectors are available to infect. Besides climate, altitude may also play a role in Japanese encephalitis transmission. Climate change may also be implicated in the expansion of Japanese encephalitis virus as higher temperatures increase the span of mosquito season in temperate regions such as Pakistan. Bird movement implicating both resident and migratory birds are associated with Japanese encephalitis virus transmission over short distances, although the mechanism involved is not well understood. Insect vectors are biting insects that may acquire and transmit agents while feeding on hosts. Without vector competent insect species, there is no agent transmission. Therefore, vector species are as important as vector host preferences, host availability, vector abundance, and vector competence, which can all be encompassed in vectorial capacity of an insect species. The environment significantly influences the abundance and distribution of vectors both geographically and temporally, seasonally, and daily. Japanese encephalitis molecular biology. Japanese encephalitis virus is a mosquito-borne virus that belongs to the family Flaviviridae and genus Flavivirus. It is a small icosahedral virus, enveloped virus of 50 nanometer diameter. It has a positive sense single-stranded RNA genome of approximately 11 kilobases. The genome has an open reading frame flanked by 5' and 3' non-coding regions. This translates into a single polyprotein which is cleaved into three structural proteins, the nucleocapsid C, membrane protein M, and envelope protein E, and seven non-structural proteins which include NS1, NS2A, NS2B, NS3, NS4A, NS4B, and NS5 by the action of viral and host proteases. Viral proteins interact with diverse host factors or machineries to complete the virus life cycle. These interactions can be cell type dependent and play a critical role in determining virus pathogenesis in a given host. The capsid protein C is a basic protein which associates with RNA to form the nucleocapsid. The envelope protein E plays an important role in viral attachment and fusion. The membrane protein M is formed from the precursor form PRM by proteinase hydrolysis leading to viral particle maturation. The non-structural proteins are an integral part of viral replication complex and interact with multiple host proteins. NS1 is a glycoprotein and is generally localized as both intracellular and secretory forms. Secreted NS1 is found in the early stages of infection in blood and is used as a diagnostic marker. NS2B is a protease complex with NS3 and is involved in processing of proteins from single viral polyprotein. NS3 is a bifunctional protein with its N-terminus having functional proteolytic domain and a C-terminus acting as a helicase. NS5 is a highly conserved bifunctional protein that consists of an N-terminal methyltransferase domain and a C-terminal RNA-dependent RNA polymerase domain. In addition to their prime role in supporting virus life cycle, viral proteins are also seen to counteract the host defense responses to support virus subsistence. NS2A protein blocks the host protein kinase R PKR mediated antiviral response, and NS5 acts as a potent inhibitor of host IFR3 and JAK-STAT signaling and antagonizes the host interferon response. Variations in NS5 have been shown to inhibit interferon alpha and beta production more efficiently in different Japanese encephalitis virus strains, giving them host adaptation advantage. NS5 is also shown to impair the host lipid metabolism, to enhance pro-inflammatory response, leading to increased neurovirulence and neuroinvasiveness in mice. Replication of the Japanese encephalitis virus starts when viral RNAs, non-structural proteins, and host factors come together to form the replication complex inside virus-induced enfoldings of the endoplasmic reticular membrane termed vesicle packets. The host cytoplasm and vesicle packets are connected through pores, which are used to translocate nucleotides and other host factors required for replication. In the replication complex, the non-structural protein NS5 interacts with the circularized positive sense RNA, and through polymerase activity of the RNA-dependent RNA polymerase domain, it synthesizes a complementary negative sense RNA, which remains base paired with the positive sense RNA to form a double-stranded intermediate. This undergoes unbinding due to the helicase activity of the non-structural protein NS3, and the negative strand serves as a template for the synthesis of a new positive sense RNA. Immediately after synthesizing a positive single-stranded RNA, a methylated cap is added to the 5' end by the methylase domain of the non-structural protein NS5. The RNA is then translocated from the replication complex to the assembly complex. In the replication complex, the non-structural protein NS1 is co-localized with NS5, and the double-stranded RNA intermediate is involved in the initial replication of the negative strands. The synthesis of double-stranded RNA intermediates and positive sense single-stranded RNAs is asymmetric, with the double-stranded RNA intermediates present at a lower copy number, and they enter again into the replication machinery for synthesis of new positive sense RNA. The positive sense RNAs are synthesized in high copy numbers and are transported to the assembly complex. The positive single-stranded RNA in the assembly complex interacts with the capsid protein for selective packaging. The final products of this replication are the double-stranded RNA intermediate and the single-stranded positive sense RNA. The double-stranded RNA enters another replication cycle, whereas the positive sense RNA is incorporated into the virion. Now let us look into the life cycle of Japanese encephalitis virus. Japanese encephalitis virus has broad tropism with its ability to infect diverse host organs and cell types. Most flaviviruses utilize the clathrin-mediated endocytotic pathway to establish infection. Japanese encephalitis virus exploits different endocytic routes, largely depending on the cell type. While infection in fibroblasts and epithelial cells takes place with the clathrin-mediated endocytosis, the virus can infect neuronal cells through clathrin-independent endocytosis. The deployment of different endocytic pathway in neuronal cells is likely to be governed by receptor usage. Replication complex biogenesis begins with the modification of the lipid composition and protein interactions on the endoplasmic reticular membranes. Virus non-structural proteins NS4A, NS4B, NS1, NS2A, NS2B, NS3, NS5, and the viral double-stranded replicative intermediate, along with other host factors, form the viral replication complex. Virus replication complexes are enriched in proteins derived from the endoplasmic reticulum-associated degradation pathway. Immature virions are assembled in the endoplasmic reticulum membrane lumen, where viral RNA genome is packaged by the C protein to form the nucleocapsid, which is further enclosed by E and PRM proteins to form the immature viral particles. These are transported to the trans-Golgi network, where the furin protease cleaves the PRM protein to the mature M protein, a step which is essential for virus maturation. The virus is then translocated outside the cell. Clinical features of Japanese encephalitis. Most Japanese encephalitis patients experience non-specific illness with acute short-lived symptoms such as headache, fever, rigor, and gastrointestinal symptoms such as nausea, anorexia, vomiting, and abdominal pain that lasts from 2 to 4 days. However, more severe Japanese encephalitis include altered senses, seizures, neurological deficit, hemiplegia, quadriplegia, or cerebellar signs, acute flaccid paralysis, and movement disorders such as dystonia of the limbs, orofacial, or axial. Prolonged convalescence is a hallmark of clinical Japanese encephalitis. Prolonged convalescence refers to the delay in recovery from the disease. In endemic areas, Japanese encephalitis may predispose patients to the Guillain-Barré syndrome, an autoimmune disease that attacks the peripheral nerves and damages their myelin insulation. About 20 to 40% of Japanese encephalitis patients die during the acute stage of the disease, with 50% of the survivors having chronic severe neurological sequelae, which include cognitive dysfunction, abnormal behavior, seizures, and movement disorders. Japanese encephalitis virus associated morbidities are primarily linked to virus entry into the central nervous system and a heightened antiviral inflammatory response in the brain, which leads to neuronal damage. Japanese encephalitis virus invasion and associated neuroinflammation. Culex mosquito transmits virus to humans. The virus initially replicates in the skin and local lymph nodes, followed by infection in the peripheral immune systems. After systemic infection, Japanese encephalitis virus reaches to the brain via newly produced Japanese encephalitis virus particles traveling through hematogenous route or by Trojan horse mechanism, that is, by migrating in infected immune cells such as the T cells or the dendritic cells. The virus infects brain microvascular endothelial cells and reaches to other blood-brain barrier associated cells via basolateral release. Successful Japanese encephalitis virus infection leads to the activation of pericytes, astrocytes, and microglial cells, leading to the subsequent release of various inflammatory cytokines and newly produced virion particles into the central nervous system. Inflammatory cytokines are said to increase the level of matrix metalloproteases and decrease the level of tight junctions in the brain microvascular endothelial cells, leading to the blood-brain barrier breach. Japanese encephalitis virus infected brain microvascular endothelial cells are reported to show enhanced level of leukocyte adhesion and chemotaxis molecules, which can result in increased leukocyte infiltration into the central nervous system, leading to heightened activation and neuroinflammation. Japanese encephalitis leads to neuronal cell death either by directly infecting the neuronal cells or by causing inflammation mediated neuronal damage, which is also referred to as the bystander. Diagnosis of Japanese encephalitis. Diagnostic confirmation of Japanese encephalitis should be based on clinical, biological, neurophysiological, and cerebral imaging findings criteria. Such findings include the bilateral hemorrhagic lesions of the thalamus that can be seen on cerebral computerized tomography (CT scan) and a characteristic of Japanese encephalitis. This is the picture of a computer tomography scan of a normal brain, and this is a CT scan that shows the accumulation of contrast material in infected areas and around the brain from encephalitis patient. Other laboratory findings include classical signs of inflammation, such as increase in polymorphonuclear leukocytes and their increased sedimentation rate, hyponatremia due to decreased secretion of antidiuretic hormone, normal or slightly increased pressure in the cerebrospinal fluid, atypical lymphocytes type 1 resulting from abnormal CD4 cells, atypical lymphocytes type 2 with typical basophilic cytoplasm and chromatin nucleus resulting from abnormal CD8 cells. Western blot, hemagglutination inhibition test, rapid complement fixation test, indirect immunofluorescence assay, and plaque reduction neutralization tests are, may lead to confusing results as they may be cross-reacting with other flaviviruses such as the dengue virus and the West Nile virus. This limits their application in diagnosis. Reverse transcription polymerase chain reaction used for viral genome amplification also allows rapid detection of RNA in the cerebrospinal fluid. However, it is rarely used. Final diagnosis is achieved by identification of IgM by ELISA in the blood or cerebrospinal fluid. It has high sensitivity and specificity and detects antibodies in 75% of the cases. ELISA method demonstrating IgM to IgG conversion in 2 successive blood samples is considered the best diagnostic tool. Treatment of Japanese encephalitis. Currently, there is no specific antiviral treatment for Japanese encephalitis that can directly target the virus. Therefore, the management of Japanese encephalitis primarily focuses on supportive care to relieve symptoms and complications. Here are the key aspects of treatment for Japanese encephalitis. Hospitalization. Severe cases of Japanese encephalitis often require hospitalization for close monitoring and medical care. This is particularly important for patients with neurological complications or those experiencing severe symptoms. Supportive care. Supportive measures are provided to manage symptoms and help the patient's body fight the infection. These may include medications for fever and pain relief. Acetaminophen, paracetamol, may be given to reduce fever and relieve headache or body aches. Non-steroidal anti-inflammatory drugs are generally avoided due to the risk of bleeding complications. Fluid management. Adequate hydration is essential, especially if the patient has high fever, vomiting, or diarrhea. Intravenous fluids may be administered if necessary. Preventive strategies. The best way to prevent Japanese encephalitis virus infection is to protect yourself from mosquito bites. Use insect repellent, wear long sleeve shirts and pants, treat clothing and gear, and get vaccinated before traveling. Intermittent draining of paddy fields and use of larvivorous fish in rice paddies are also control methods that have been applied in affected areas. Another prevention strategy that has been debated is vaccination of pigs. Though theoretically it could represent a strategy to target the main amplifying host, it is not cost effective in most settings due to the high pig turnover in industrial farming. Vaccines for Japanese encephalitis. The mouse brain derived Nakayama and Beijing 1 Japanese encephalitis viral strains were purified and formalin inactivated to produce the first Japanese encephalitis vaccine, JVAX. The efficacy of JVAX is above 90%, but it has high cost, lacks long-term immunity, and shows allergic effect and was discontinued in 2005. The LAV vaccine was generated using multiple passages of wild-type SA14 strain in animals and cell culture. Purified, formalin inactivated whole virus vaccine IC51, IXIARO, or JESPECT based on the attenuated SA1442 strain cultured in Vero cells is licensed in USA, UK, Canada, Australia, and Europe. A live attenuated vaccine JEV SA 1442 cultured in primary hamster kidney is licensed for use in China, Korea, Cambodia, India, Sri Lanka, and Nepal. Despite the excellent efficacy, 85 to 99% of SA-1414-2 vaccine in Nepal, China, and South Korea, the efficacy of the same vaccine in Indian adults was found to be lower. The only genetically engineered licensed vaccine is the Japanese encephalitis chimeric virus vaccine, JECV, or chimeric vax JE, or Imojev, is available in China, Thailand, Australia, and India since 2010. JECV was designed to be attenuated, in which the precursor M and E genes of yellow fever virus 17D vaccine strain were replaced by the respective genes of Japanese encephalitis virus. The latest Japanese encephalitis vaccine, Genvac, was developed by National Institute of Virology, Pune, India, in collaboration with Bharat Biotech International in 2013. The JEB strain Kolar A21564XY was adapted and inactivated in vero cells to produce the Genvac. This vaccine can be given to age group of 0 to 50 years. Challenges associated with Japanese encephalitis prevention. Mosquito control is crucial for Japanese encephalitis prevention, but it is particularly challenging as the porcine and avian reservoirs of Japanese encephalitis virus are the key sustaining and amplifying factors of the disease. Japanese encephalitis vaccines need to evolve to provide protection against the newly emerging genotypes and also be able to provide long-lasting immunity. Several of the Japanese encephalitis epidemics occur in low-income rural settings, and it is likely that malnutrition could be a contributing factor to low vaccine efficacy and poor disease prognosis. Another crucial aspect of Japanese encephalitis pathogenesis that needs attention is the development of novel therapies for management of a heightened and uncontrolled neuroinflammatory response. Now, a short summary. Japanese encephalitis virus is the most deadliest viral encephalitis in Asia, which is caused due to the vector Culex mosquitoes infected with the Japanese encephalitis virus. The birds serve as reservoir hosts, pigs serve as amplifying hosts, and humans as dead end hosts for these viruses. Japanese encephalitis virus belongs to Flaviviridae and is a closest relative of the dengue virus and the West Nile virus. It is mostly observed in the rural areas of Southeast Asia, the Pacific Islands, and the Far East. Symptoms of this disease include high fever, headache, neck stiffness, seizures, coma, spastic paralysis, and ultimately death. Treatment. No antiviral drug therapy is available for Japanese encephalitis except the palliative care. The patients are hospitalized during the infection and are closely monitored. Preventive measures. Vaccination with the live attenuated SA-1414-2 vaccine and avoiding mosquito bites. These are a few papers for further reading. Now, let us discuss some multiple choice questions. What is the primary mode of transmission of Japanese encephalitis virus? A. Airborne. B. Vector-borne. C. Waterborne. D. Direct contact. The answer is B. Vector-borne. Which of the following is a natural reservoir of Japanese encephalitis virus? Pig, birds, rodents, monkeys. A. Pigs. What is the primary vector responsible for transmitting Japanese encephalitis virus to humans? Aedes aegypti, Anopheles mosquitoes, Culex mosquitoes, Ixodes ticks. The answer is Culex mosquitoes. Which organ is primarily affected by Japanese encephalitis? A. Liver, B. Brain, C. Kidneys, D. Lungs. The answer is B. Brain. Which of the following is a common symptom of Japanese encephalitis infection? Skin rash, joint pain, enlarged spleen, severe headache. The answer is D. Severe headache. What is the most effective method for preventing Japanese encephalitis? A. Antibiotic treatment. B. Vaccination. C. Use of mosquito nets. D. Quarantine measures. Answer is B. Vaccination. Thank you for listening.